I'm Masayo Komoda, Department of Pharmacy, Chiba-Nishi General Hospital, and Faculty of Pharmaceutical Sciences, Tokyo University of Science. My title, Ivermectin via unique whole body basing for elderly patients with scabies. I worked as a pharmacist in a geriatric hospital in Chiba, next to Tokyo in Japan. The hospital where I worked experienced a scabby outbreak in 2000. One inpatient was diagnosed with crushed scabies, and all 33 patients and 18 staff on that ward were found to be infected a week later. The situation was miserable. That the ward was isolated and the scabies patients were cared by infected staff. At the time, the only effective drug available for scabies was the experimental reagent gamma PHC. As a pharmacist, I had a difficult time with the use of gamma PHC, which is already banned as a pesticide. I was very upset that there was no effective and safe treatment for scabies in Japan. And together with dermatologists, we requested the approval of topical permethrin or oral ivermectin, which were used abroad. In 2006, I enjoyed the faculty at my alma mater, the Faculty of Pharmaceutical Sciences of Tokyo University of Science. Then I started a clinical research project with a dermatologist to solve the problem of scabies treatment. I present my research as follows. What is scabies? A skin infection is caused by a parasitic infestation of the skin by very small mites. You can see photo 1. Since the mites do not suck blood, the patient is only aware of the infection when a rash and itching appear after a latent period of about 1 month. Photo 2. The definitive diagnosis is detection of the mites, but they are difficult to find and the diagnosis is delayed. If steroid treatment is continued without noticing scabies, the number of mites, 1 million or more, may develop abnormally, resulting in crushed scabies. See photo 3. The crushed scabies is highly infective and can cause mass infection. In some cases, medical practitioners may serve as the carriers of infection. We got an approval of oral ivermectin in 2006. Trade name is Stromectol. Subsequently, in 2014, topical phenolphthalein, which is the same pyrethroid family of permethrin, and was widely used as a pesticide in Japan, as approved. Trade name is Smithline lotion. 2 drugs for scabies are currently available in Japan. Do you know who discovered ivermectin? Dr. Satoshi Omura discovered ivermectin from Japanese soil. The Nobel Prize was awarded to Dr. Satoshi Omura in 2015 in recognition of the fact that ivermectin saved 200 million people from river blindness. Different mechanism of action of ivermectin in the treatment of river blindness and scabies. Microfilariae, the cause of river blindness, enter human skin via nuts. They then become adults in the body and lay thousands of eggs, which are released into the bloodstream throughout the body, repeating the infection cycle. Therefore, to be effective against river blindness, ivermectin needs to enter the blood circulation. On the other hand, the mite that cause scabies do not migrate into the bloodstream and only need to reach the skin surface and the stratum corneum. Next problem with scabies treatment. The two treatments have their own advantages and disadvantages. Ivermectin oral application is simple, but has systemic side effects and cannot be used by pregnant women or infants. On the other hand, topical phenytoin is safer, but require more effort for caregivers to apply all over the body and recover from missed application. Next, research into the practical application of unique ivermectin whole body basing. Haas et al. Reported skin stratum corneum concentrations of 40 to 80 nanogram per gram in patients who were effective after oral ivermectin. Ivermectin superior in that it is effect at very low dose. One of my collaborators, lecturer Miyajima, an expert in pharmacokinetics, said, for stromectol tablets taken by an adult at one time in a typical Japanese household bathtub on approximately 150 liters would produce an ivermectin solution of 80 nanogram per milliliter. When I asked him why, he said, could immersing patient in an effective ivermectin solution that can eliminate the mites make for a complete treatment? I said, I see, great idea. I was really excited. Economical if several people take the same bath. It's named Ivermectin whole body basing. The method has the potential to solve all of the following problems. This method can be definitely applied to the whole body from the neck down. Ivermectin has low skin permeability, so systemic side effects may be avoided. Describes the influence of different molecular weights on skin permeability. The molecular weight of ivermectin is 155, larger than phenytoin, so 350. When the molecular weight exceeds 500, permeability through normal skin is reduced unless the skin barrier is destroyed. Therefore, ivermectin is difficult to penetrate through the skin. The concept is based on above. In ivermectin whole body basing treatment, ivermectin does not enter the bloodstream, but reach effective concentrations in stratum corneum. The concept was first confirmed in animal experiments. The research started in 2011, firstly by confirming the concept in animal experiment. Next, the optimal condition for ivermectin basing solution was studied in healthy volunteers, and the safety and feasibility of ivermectin whole body basing were confirmed. Finally, safety and effectiveness were confirmed in patients. The above study took 10 years. Animal experiment. Ivermectin basing solution was prepared, and rats were based for 5, 15, and 30 minutes, and blood and skin ivermectin concentration were measured. Results showed that ivermectin was detected in the back and abdomen, but not in the blood. These results confirm the concept of whole body basing, as ivermectin is detected on the skin, but not in the blood. Next, a partial bus test was conducted on healthy volunteers to determine the optimal condition. Warm water with stromectol tablet was prepared and compared with and without baby ablution. The result showed that the stratum corneum concentrations of ivermectin were all higher than the exceed effective concentration. The variation in ivermectin concentration in the stratum corneum was smaller with the use of baby ablution additive. As ivermectin is insoluble in water, the addition of the ablution additive was thought to improve its solubility and make it a more homogeneous solution. Based on the above, ivermectin whole body basing was detected in surface amount of ivermectin in the stratum corneum in humans, and the addition of ablution additive reduced the variation. Then, safety and feasibility were examined in healthy volunteers. 6 healthy volunteers were bathed in the bath for 5 minutes. After bathing, they were wiped lightly with a towel, and the stratum corneum of their palm and upper arms were sampled 1, 4, 8, and 24 hours after bathing. Samples were also taken 1 hour after bathing and 1 week later to measure ivermectin concentrations and to perform blood chemistry tests. In all 6 healthy adult volunteers, ivermectin in plasma was below the limit of quantification. In addition, ivermectin concentrations in the stratum corneum of the forearm and palms were all significantly higher than the concentrations considered effective up to 8 hours after bathing. No problematic skin symptoms, subjective symptoms, or laboratory abnormality were reported. In conclusion, the ivermectin whole body basing was considered to be well tolerated. potentially effective and feasible. Clinical trials were then conducted to confirm effectiveness and safety in patients with scabies. The facility where scabies had infested were visited, and then ivermectin whole body basing were given with stromectol tablets in the bath. The process was repeated once a week until the mites were undetectable for 2 consecutive sessions. The effectiveness was assessed by examining the mites, monitoring the extent of itching and skin rash, and the safety was assessed by blood biochemistry tests and observation of skin symptoms. When feasible blood sample and stratum corneum sample was also taken 1 to 4 hours after basing to measure ivermectin concentrations. It took 5 years for 7 patients to participate in the study, as scabies is a disease that can occur anywhere at any time. The average age of the 7 patients was 82.7 years, with 2 males and 5 females. 5 of the patients required caregiver level 5 and were bedridden residents, while 2 were users of the facility day service and were bathed at the facility. 1 had crushed scabies, and 6 had normal scabies. The result shows that the average number of ivermectin whole body basing was 3.1, and the average time to cure was 23 days. The ivermectin concentration in 1 case of stratum corneum sampling was well above 80 nanogram per gram. No adverse events were observed. Conclusion. The ivermectin whole body basing was effectiveness for patients with scabies. No adverse events were observed. The stratum corneum concentration of ivermectin in 1 cooperating patient was significantly higher than the effective concentration when taken initially, and no ivermectin was detected in the blood. With hope for ivermectin whole body basing. Based on these results, more than 20 years after the scabies outbreak experienced in 2000, outbreak continued to occur. And based on these results, the ivermectin whole body basing proposed in this study can be implemented for the treatment of scabies and is safer than oral administration, considering the systemic exposure level of ivermectin. In countries where basing is common, such as in Japan, ivermectin whole body basing should contribute to the early resolution of scabies outbreaks in facilities if ivermectin whole body basing is performed on both patients and staff at one time. Thank you for your attention. This research is a result of the efforts of the students of Komoda's laboratory, Faculty of Pharmaceutical Science, Tokyo University of Science. The doctors in charge of the home the dermatologists, the nurses, and the nurse care staff. We would like to thank them from the bottom of our hearts. Thank you.